Right, so in the miscellaneous folder or PowerPoint, I guess you could say, there are a couple different chapters we kind of jump around in. And so I'm going to start with chapter 13. Chapter 13 is really kind of important in that it's going to explain the lab we're going to do tonight. So I'm going to start by talking about just the general premise of evolution, and then tonight we're going to try to do a lot of complicated math without any additional support, because I was really hoping that they were going to do another video for you and give us data, but I have gotten nothing. So we will make this work somehow, me and my little fancy schmancy whiteboards. So we'll do the best we can, but we will try to get through it together. So let's first just talk about the concept that this lab is going to be about. So first of all, we always talk about Darwin and his journey. And so when we talk about his theory, what we knew is that there were a lot of different scientists who were talking about this idea of evolution, this theory of evolution, but we have I gotta deal with dogs now. Okay, the dogs have been kicked out, and I have to lock them out because my German Shepherd opens the doors. So they are locked out. So we can finish this hopefully with no further interruptions. So like I was saying, there were many different things or many different concepts or ideas that we were making these observations, and they were leading to the theory of evolution, but we didn't have a mechanism for them, like the how. How did evolution occur? And that is what Darwin gave us, was the how. So we know Darwin went for this voyage, and I don't want to get too far ahead of myself because I think we have it in a further slide. But this voyage, this 5-year voyage that Darwin went on, he made tons of observations, many specimens. Some of his, this is the blue-footed booby that he did a lot of observations on. But he's really famous for many of his observations made at the Galapagos Islands, which were a fairly new island chain, a volcanic island chain. And so you could actually see how the populations came to be on this island and how they evolved and adapted to each of the island's different environments. And so if we talk about some of the earlier concepts, we have talked a little bit about them in another PowerPoint. So when we talk about them, we know that some Greek philosophers suggested that life might have changed gradually over time. However, the Greek philosopher Aristotle, he viewed species as perfect and unchanging. And then we have the Judeo-Christian culture, which reinforced this idea with the literal interpretation of the biblical book of Genesis. And again, if all of these organisms were created by God, they would be perfect, and so they wouldn't change. And that's not what we see. It's not what we see with fossils. It's not what we see with different specimens. And so with those fossils and those different specimens, we do make observations that species do change over time. They have evolved. But again, the question is, well, how? Well, poor Lamarck. We are always making fun of Lamarck because this is one of the, no, this is not right. So in 1800s, one of the scientists that proposed or had a mechanism that he proposed for how evolution occurred was with giraffes. And so what he said was that these giraffes, as they ate the leaves on the lower branches, the leaves on the higher branches would be available for eating. And so as the giraffe stretched and stretched to reach the higher leaves, the neck stretched. And then this acquired trait, so as this giraffe obtained this long neck from stretching, this would be the acquired trait. It would be like if you put ear gauges in your earlobes, and you know how they get bigger and bigger, which I personally don't like them. They kind of creep me out. But that would be an acquired trait, something that you gained during your lifetime. But what Lamarck proposed was that then when you had a baby, it would also have holes in its earlobes. And so when this giraffe that had this long neck had baby giraffes, they also had long necks. And so that was his idea, which we know is absolutely ridiculous. And so that was one of the bad ideas or suggestions. So again, we always talk about the breakthroughs, but what about all the things that weren't um, weren't necessarily correct? Okay, so which we have quite a few of those if you look into the history of some of the science concepts. So during Darwin's round the world voyage, so he was, so basically what Darwin did was Darwin went to medical school because his dad and his grandfather, they were all doctors, they were all physicians. And when it came time for dissecting, yeah, he was not very fond of blood. And he said, no, this is not for me. So he went into the natural sciences, and he had a friend who actually knew that the captain of the HMS Beagle was looking for a science officer, and he recommended Darwin for this position. And so Darwin took this position. It was a 5-year voyage, so he took tons of books with him because we didn't have computers and stuff like that back then. And so with those books, one of his more famous books that he actually used to come up with this idea was Lyell's Principles of Geology. And what we were finding, again, if you look at the Judeo-Christian concepts, the earth is only, I don't know, a couple thousand years old. And so what Lyell was showing with geology is that many of these structures that we see on earth took millions of years to develop. So like the Grand Canyon was not carved in a couple thousand years. It took millions of years. Mountain ranges, millions of years of uplifting. So he was saying that the earth has gradually changed and is very, very old. In fact, it's billions of years old. And so Darwin then started to think, well, if the earth is really old, well, then the living organisms on it must also be really old. And so we have this change. So again, today, present day species had to have arisen from ancestral species by natural processes. So what were those natural processes? And so we had to try to figure out, well, what's the mechanism then? What is this process that allowed these organisms to change over time? And so some of the organisms that he is famous for, so besides the blue-footed booby, is we have the marine iguana and the Galapagos tortoises. So like I said, he collected many different specimens, made tons of observations during his voyage. And here's a picture of that voyage. So you can see he traveled a great distance. So he started in England. They went all the way down to South America, around South America, through the Galapagos Islands, and then popped out over there in New Zealand, Australia. They didn't really go to, I don't think they went to Madagascar. The thing doesn't go to Madagascar. It would have been really cool if Darwin had gone to Madagascar. We know there's so many different things there. And then back to South America, and then back up to England. So he was gone for a long time on this trip. So in 1859, Darwin actually published his book on the origin of species by means of natural selection. Now, it was really kind of interesting because he actually told his wife upon his death to publish his book because he knew it was going to cause a ton of controversy. And so what happened was a friend of his, Henslow, actually knew that another man, Wallace, had come up with the same conclusions. And although he didn't have nearly the support Darwin did, he told Darwin, he's like, you need to publish because, or you're going to be scooped. If you watch the thing with Watson, he talked about being scooped on his double helix idea or model. So if you did not take time to watch that TED talk on Watson, it is really funny. But scoop meaning that someone else is going to beat you to the punchline here and take credit for this novel idea. And so Wallace and Darwin actually agreed to publish at the same time. we don't hear much about Wallace, although they both did come up with this idea of natural selection. We hear so much about Darwin because Darwin just had volumes of evidence to support his descent with modification, meaning that each generation had some change to it. And the way that change came about is by natural selection. So again, he noted many organisms over various different habitats, and just so much information that he had to support his theory. So like I said, Darwin devoted, I think he spent almost 20 years after he got back looking at, I mean, he was just sending specimens to all these warehouses back in England. He had plenty of stuff to study while he went through. Darwin also discussed many examples of artificial selection in which humans have modified species through selection and breeding. And we can look at that. I mean, you think about all the dogs today are related to a wolf. That is the original dog. And think about how we have selected certain characteristics that we have desired from that population of wolves to give us all the different dog breeds that we have today. Kind of crazy. So that is an ideal. And we don't just do it with dogs, we do it with cats, we do it with horses, right? We're totally always breeding horses to be successful and for Triple Crown winners or whatnot for horse racing in that arena. So you could look at that again as evolution basically on steroids. So that is another example of evolution. We also talk about antibiotic resistance. That is another form of evolution. We get a lot of times where people say that evolution is so slow we are not able to witness it in our lifetime. I've just given you several. I mean, we've done dog breeds. It's only been about 100 years that we've been modifying and changing these breeds. And antibiotics, well, we see that in our lifetime all the time where a bacterial cell is no longer affected by a certain type of antibiotic. It's kind of scary how fast they are evolving. So Darwin recognized the connection between natural selection and the capacity of organisms to over reproduce. So he actually read an essay written in 1798 by economist Thomas Malthus. And he argued that human suffering was the consequence of human populations increasing faster than essential resources. So what he is saying is basically there are more people than we can support. And that's also what Darwin noticed, that in an environment, there are generally more organisms than that environment can support. Therefore, you get competition for those resources. That competition, those individuals that have the best set of adaptations or characteristics will be more successful, and they will out reproduce those individuals that do not have those favorable adaptations. And so you get a shift in the population where more individuals have that favorable trait than not. And so this is evolution. And it's not necessarily, which many of you guys think of, oh, one species changing into another species. No, we are simply looking at the change in an adaptation, like human evolution. You look at height, human height has changed. We are getting taller. Our average height is taller than it was 100 years ago. That is evolution. It is on a micro scale. We refer to it as microevolution. And that is where we have gene frequencies. So gene frequencies, and that's where I'm going to start throwing these terms in for our lab. That means how often does that gene show up in the population? Well, right now we have more tall genes than we have short in the past. Okay, so it has shifted. So what Malthus said was, we don't have people that are, just because they're taller, they're surviving. But what we can say is the things that have kept human population in check is war, famine, and disease. So those three factors have kept the human population balanced. Right? So that's what Malthus's essay was about. So Darwin like I said, he observed this in organisms. They vary in traits. And so we want varying traits in a population. We want variation in a population. When we do not have variation, this is where we lead to problems. This is where if an environment changes, that population may not have the ability to cope, to survive it, and the whole population dies. That is a problem. So we want the more variation, the better, which is why when we have these endangered species, we start reducing the gene frequencies of certain adaptations, and it's a problem. Okay, they are not able to adapt to changing environments, which we know our environments are constantly changing, and they are not successful and they die. He also observed the same thing. There are more offspring in an environment than can support it, and that leads to competition. And this is where we always talk about survival of the fittest. It is that competition. It is not a matter of who can do more sit-ups or push-ups. It is a matter of who has the right traits that makes them successful, that they are able. And when we measure success, what our measure of success is producing offspring, viable, fertile offspring. That success is reproductive success. That's what we're talking about, is passing those genes on to the next generation. So Darwin reasoned that organisms with traits that increase their chance of surviving and reproducing in their environment tend to leave more offspring than others. And so we get what's called unequal reproduction, and it will lead to an accumulation of favorable traits. As we start to do that, we have now shifted our gene frequencies, and this has changed the population. This is evolution. This is microevolution. And that is kind of what we're focusing on, not macro. Macro is, yeah, we're completely new species. We're talking about microevolution here. There are 3 key points about evolution by natural selection that clarify this process. Okay, so number 1, individuals do not evolve. Populations evolve. I will say that over and over again. Well, I would be saying that over and over again if we were together, but I'll say it again. Individuals do not evolve. The population evolves. Natural selection can amplify or diminish only heritable traits, not acquired traits. You cannot pass on acquired traits. Evolution is not goal-directed. It does not lead to perfection. Favorable traits vary as environments change. So what may have let that population be, that adaptation, that trait that allowed them to be favorable in that environment, if the environment changes, so maybe they do well and when there's a drought, but now there's no drought and maybe there is flooding. Maybe that trait does not allow them to be successful with the flooding. So there's no goal, because there's never, it'd be like you're constantly moving that goal. You can never reach it, because we know our planet is always changing. The environments change. Scientists can observe natural selection in action. So we always like talking about camouflage, because camouflage is an adaptation in insects, and it has evolved differently in different environments. And so when we look at these praying mantises, and you can actually, most of you should know what a praying mantis looks like around here. We have quite a few of them here in Illinois, but you can see how these two are very different. They're both species of praying mantis, but one is in Malaysia, where we have this flowering plant. And so if you were an insect that you resembled that flowering plant, you wouldn't be picked off by predators. And so again, you'd be able to successfully reproduce and pass, and your offspring would resemble you, okay? If not, they would get eaten. And then maybe some of your offspring resembled you, and they would be successful and then pass on those traits. And so eventually we would get more and more praying mantis that look like this flower. Versus Costa Rica, we have these broad leafy plants. And so this body shape or adaptation or characteristic or trait, whatever you want to call it, or allele, right? Allele is another one. It's the gene, the version of a gene. So this version of looking like a green leafy plant was more successful for this praying mantis in Costa Rica. So biologists have documented natural selection in action in thousands of scientific studies. We have Rosemary and Peter Grant are famous for their research on Darwin's finches in the Galapagos Islands, again, because people would say, oh, it's too slow, evolution is too slow for us to see in our lifetime. Well, here they have been noticing these microevolutions and these finches now for 30 years. They've been looking at it. So in wet years, we have small seeds on that island are more abundant. So if you have a smaller beak, you're able to manipulate and crack those seeds open and eat them very well. If you have a large beak, you can't quite get the food. Okay, maybe some of you guys understand that if you've had boyfriends or husbands. Um, or maybe Eric, right? So you've had something you need really fine little fingers for, and maybe you've had to ask a female to help you out with that because we have much smaller um, fingers. And so you're trying to manipulate these little tiny seeds, it's not going to work. Right. So the birds with the smaller beaks are much more successful. They have more food, so they can reproduce and have more offspring. And those offspring, again, will hopefully be just as successful as them because they have smaller beaks. Now, in dry years, because we didn't kill off all the large beak finches, they're still there. They're just not doing very well. All right. So now let's say the island has a dry year. Now in this dry year, the large beaks are favorable because all seeds are in short supply. And so the birds have to eat the larger seeds. And if you have a small beak, as you know, as females, there's things, I can't open that jar, right? I don't have big enough, strong enough hands. So I got to ask that male with the big, strong hands to open up that jar. And so the birds that have the bigger beaks are like, no problem. We got the bigger seeds that we can use. So again, the population is being successful by having this variation. The individual, the small in the different environments, the small or the large beaked bird, they're SOL, right? But their population is being successful. Another example of natural selection is the evolution of pesticide resistance in insects. So you may have a relatively small amount of a new pesticide. So you have this new pesticide, it kills 99% of the insects, but that 1% that survive, they reproduce and they reproduce. And guess what? Their offspring may also have that insect, that pesticide resistance. And so again, you spray the field again, killing off the weak and leaving those individuals that have that mutation that allows them to be resistant. And again, they are living to reproduce more bugs. So in this picture, we can see here, we have now the green and the red are representing different alleles, right? So the allele, the red allele are the survivors. So those that have the red allele or the red version of that gene happen to survive the pesticide. So we're killing off all the green ones. Now we have the red ones, and now we have all red left. Now, remember your genetics. We're only showing one gene here. So they could be heterozygous, meaning that there could still be some carrying the green gene. Don't know, but I want you to think about that. You know, to keep that variation. Heterozygous individuals are really can be important in maintaining genetic variation within a population, which, like I said, is really important. So these examples of evolutionary adaptation highlight two important points about natural selection. One, natural selection is more of an editing process than a creative mechanism, right? So it's there, it's either a mutation that has occurred, it's been sitting there maybe dormant and hasn't caused anything, you would never know until the situation arises. Got a new pesticide, guess what? I happen to have that gene. Okay, so it's not like, oh, we have a new pesticide, I need to make a new gene. No, it's there. And if it's not there, they would have all died. The whole population would have died. And we see that with species going extinct, right? They don't have the right combination of genes, they can't survive. It's also, like I said, natural selection is contingent on time and place, favoring those characteristics in a population that fit the current local environment. Right? So it's that habitat that that organism is in. Darwin's idea about evolution also relied on fossil records. We know the sequence in which the fossils appear within the strata of sedimentary rock, so we can look at that. We can see the older fossils are lower in the sedimentary rock. The newer species are on top. Paleontologists study these fossils all the time. They give us a lot of information about the anatomy, physiology, and the time period in which those organisms lived on our planet. Fossil record also shows that organisms have evolved in a historical sequence. The oldest known fossils go back 3.5 billion years ago, and that would be our prokaryotes, so our bacteria. The oldest eukaryotic fossil are about a billion years younger. So it took another billion years before we got eukaryotic cells. Another billion years before we find multicellular eukaryotic life. So it took a long time to get life jump-started on this planet. Many fossils link early extinct species with species living today. So we have a series of fossils that traces that gradual modification. So we can see actually in animals, we start off with jawless fish, and then we go to our jawed fish, and we move on to our amphibians and then reptiles. And eventually, once we get to our reptile ancestors, we can then, that kind of branches off into, it's kind of cool, branches off into like dinosaurs, reptiles, and reptilian like mammals, which lead to the mammalian line. So really kind of interesting. We can see here, we have a series of evolution of whales. You know, life started in the water. So we started with our fish, amphibians, then left, you know, the water, but we're still tied to the water for laying eggs. And also they had to keep their skin moist. They would dry out. And then we moved to adaptations that allowed their skin not to dry out with scales, and an amniotic egg that we see that has a protective leathering coating on it, which we have our reptiles. And so life started in water, it then transitioned, right? Could be on land some of the time, and then it all went to land. And so then it's kind of interesting that once mammals, that whole group of organisms, the mammals, were on land, some of them went back to the water. And we can see this with a whale fossil. So here is our terrestrial ancestor of a whale. It then returned back to the water. And so you could hypothesize that those individuals that had reduced back limbs were able to swim better, maybe stay away from predation or hunt food. And so as that reduced further, we can see we have our today's whale. So this is our recent whale. And with that recent whale, we can see here, this is the remnants of a pelvic girdle and a hind limb. We call these structures vestigial structures. So vestigial structures are things that we see that are basically the remains of, and they're usually reduced in size and don't function anymore. But they are kind of the remnants of a structure that at one time did have a function. So it's kind of interesting. You would think, why would a whale need a pelvic girdle and hind limbs? Well, because of originally they came from ancestors that were on land. And then we get into this thing called biogeography, so life based on the land. So we talk about geographic distribution of species. And so we know that species come from a common ancestor. He noted that the Galapagos animals resembled, so if we look at those, like I said, those were kind of, you know, whether they're 2 or 3 million years old, which is a baby on our planet, but the organisms resembled the organisms on the mainland, so South America. So somehow their ancestors migrated, they got to those islands and they got stuck there. And then they evolved differently because the populations are now separated. So they have a different set of skills that they may need in that different environment. And so as the years progressed, they became more and more different than the mainland animals. Some of the things that we use to support evolution is comparative anatomy. We can compare many of the body structures of different species, and again, we can see some of those similar structures. When those structures come from the same body parts, we call them homologous structures. And so I like to show this picture in general. What a homologous structure is that it's from the same body part, it's from the same structure. They're organized the same, but they have a different function. And so when we look at this picture here, we can follow these bones, and you'll be doing all this anatomy and physiology when you take that class. You can see they all have a humerus. Then they all have a radius and an ulna, which are the two bones in the lower limb. And then we have what are called the carpals. Those are the bones in the wrist. And then we have the metacarpals, which are the bones in your hand. And then you have your phalanges, which are the fingers, the digits here. So we can see these in all these mammals here, but they have different functions. So like humans, we use our hands for grasping and using tools. Cats for jumping and running and capturing prey. Whales for swimming, and bats for flying. So same structure, they have been modified for a different function. So again, evolution doesn't start from scratch. It takes what it already has to work with and modifies from there. It's more of like a tinkering process. It may tinker in a negative way, and if it does, that individual will then die and it won't be so helpful for the population. If that tinkering is positive, that individual will be successful and pass that, hopefully, if it is heritable, right? It has to be heritable, pass that on to their children, and then we will start to get a shift in the population with that gene frequency. We can also, and actually Erasmus Darwin, that would be Darwin's grandfather, he did a lot of comparative embryology. And so we can look at the stages of these embryos, and a lot of the embryos actually start off looking very similar, and you wouldn't be able to tell the difference between a human, a pig, a monkey, those embryos at all at the beginning. It isn't until they get further along in their development where they start to diverge and take different pathways. So it's almost like they have the same set of instructions for days 1 through 30 or whatever. And then we have another different, okay, now we got to go to this different direction. And so we still see those homologies. Some of the homologies, like I said, in the arms and stuff in the adult organisms, but some of the things that all vertebrates, so all vertebrate embryos, that is, they have a backbone. They all have what's called a posterior tail. So that means past the anus, there is a tail. And you can actually see that in our embryos that we had what's called post-anal tail. It is absorbed back into our body, kind of like a tadpole, right? Tadpole has a tail and then it gets absorbed. It doesn't fall off, it gets reabsorbed. It undergoes apoptosis, and those tissues get reabsorbed and broken down. And then we have our remnants of that postanal tail with our tailbone, right? And we also have what's called the pharyngeal gill slits. And so those pharyngeal gill slits, and we do have a picture here in the next slide. So I'll wait and talk about that in the next slide. I already talked about vestigial structures, right? Remnants of features that served an important function in the ancestor of an organism. And then here is that picture. So we have that postanal tail. So here's our human. See right there, there's our postanal tail. This is a little chick versus a human. And then we have what's called the pharyngeal pouches, so, or gill slits. And they do form gills in our fish and other types of aquatic organisms that are vertebrates. But for us, and actually in a lot of the jawed fish, these pharyngeal gill slits actually formed parts of the inner ear in our jaws and our throat structures in humans. So those structures were modified for different purposes. They were repurposed for different functions. Okay, so we see these in all our vertebrates in this embryonic stage. And there's another way that we can actually compare organisms, and that is by molecular biology. So we can look at the DNA. And not only can we look at the DNA, because sometimes the DNA has some mutations, but even with those mutations, we still have this same amino acid sequence, right? We talked about those silent mutations. So whether we look at the DNA or the amino acids and the proteins, we can study multiple species and compare them. And what we do note is there are some genes that we find in almost every species. And so we can see how life forms are related. We can see how they all share common DNA that codes for proteins found in the living cells. So this kind of brings us back around to what we've been studying all year around, long. We know that, like I said, humans and bacteria even have some homologous genes that have been inherited. So, we get in that whole debate, are we related to chimpanzees? We have genes that are found in bacterial cells. So, I don't like that question. Are we related to chimps? Well, we have a common ancestor to chimps. Meaning, and if we compare our DNA, we're like 98 or 99, I don't know exactly, percent the same. Very, very closely related genetically. Okay, but we are not a direct descendant from chimps. We have a far, far common ancestor. Okay, so if you take, and I do recommend an anthropology class, they get into that. Physical anthropology is a great class if you're interested in looking at the evolution of humans. Darwin was the first to represent the history of life as a tree. So with multiple branches from a common ancestral with a trunk. And he also, when we do this tree, we have these ends, branches that have ended in extinction, and then other branches that have diverged. So today we call this an evolutionary tree. Oftentimes we turn it sideways, and I'm going to show you that tree here in the middle. And what we generally do is we need to use homologous structures. We use structures that came from the same body part. Okay, so like for instance, we don't really talk about analogous, but there is the opposite of homologous, and that is analogous. And so when we talk about a homologous structure, again, same structure, same body part, different function. We use it differently. An analogous is the opposite. It is a different body part, but used for the same function. So for instance, a butterfly's wings and a bird's wings. The butterfly wings is an outgrowth of the exoskeleton. And a bird wings have to do with, and you kind of saw that with the homologous structures, are modified forelimbs. There are not bones in a butterfly wing. So if you can't picture the homologous structure of the bone slide I showed you, but you do know there's no bones in a butterfly wings. So they solved a problem of evading predators or finding food by flight, okay, by having wings. But they solved it differently. So they have the same outcome just by a different method. That's analogous. Now, we don't want to compare when we're looking at evolutionary relationships. They're not related. Those wing structures are not related. So we're not going to look at analogous structures. We're only going to look at homologous structures. Okay, so here is a picture of that tree on its side. We generally call these things, they can be called a cladogram. So what we're going to do is we're going to look at them, and we can see how the lungfish are distant from anything else, right? They're further back. We finally get what's called the tetrapods. Now, believe it or not, snakes actually had 4 limbs. That their limbs were, like the whales, were reduced in size, and we actually have what are called claspers, which are kind of the remains of that back pelvic girdle that's actually kind of been repurposed. It actually helps male snakes to grasp on and copulate with the female. So we have our tetrapod limbs. So everything on this side of that branch has 4 limbs, or had an ancestor that had 4 limbs. And then we have the amnion, which is the amniotic egg. So these structures, that allowed them to either lay eggs on the land, or we talk about mammals, right? Mammals have their placental, so we take that a little bit further. And then they kind of separated out based on feathers. So there are different ways, and depending on what traits you're looking at, that you can kind of organize this. It's really kind of interesting, because when you look at crocodiles, they're constantly trying to decide where to put it, in with the birds or in with the reptiles. So if you find that fascinating, you could look that up, and you'll see there's different, depending on the different schools of taxonomy. So we don't have even an agreement with our taxonomists, which are, there's different schools of taxonomy, which I'm not going to get into in this course. And so that is all that I'm going to cover today. The rest of the chapter, you're going to want to read 13.7, 13 through point 11. That is going to help you with your lab material. So do make sure you read those sections. Ideally, you would have read them before the lab today. Hopefully, you will read it, try to read it before you start asking me for questions, because it might make sense by reading it in the book with the pictures and all that stuff. So that is where I'm going to end the lecture for today. And then what we're going to do next is we're going to do the lab. All right.